All good. Okay. Hi, everyone. Good morning. Welcome to the Metabolomics Summer Course 2022. I'm Effie, and I'm a PhD student like a lot of people in here. I'm a PhD in Copenhagen, actually, and I know that I flew with quite a few people from Copenhagen. Cool. So today will be a day of theory in general, so bear with us. I think me, Martin, and Daniel will be presenting experimental design, QA/QC, MS data acquisition, and some projects that we have, some really interesting projects. Okay, so we're starting with experimental design. So generally, an experiment can be hypothesis-driven or data-driven, as you know, and normally we aim to answer the question about what is the difference, what is the effect of a process or a treatment within different groups of subjects. And the goal of the experimental design is to minimize any technical, experimental, or biological variance that you have during the whole process. And of course, provide valid results, ethical results, and minimize your expenses, right? So some examples when you work with metabolites is that they're temperature sensitive, so you need to be careful with the storage conditions. They might be light sensitive, so you need to be careful with UV light coming from your window in the lab. And you have to have consistent growth conditions in general, so you can reproduce your data. And of course, technical biological replicates and blanks that we will thoroughly talk about towards the end of the presentation. And also Martin will talk a bit about that. Right, so the key aspects of the experimental design is the scope of your study. You need to make sure that you know what you're doing and what the goal is. Maybe pre-calculate your sample size before you go into the lab. And of course, randomization that I'm not going to go through on detail here. I'm sure that some of you are working with clinical metabolomics, which is randomization is a bit more complicated there. So let's say you have two different groups that you want to compare the effect of a vaccine, right? So you have group A, that's control, and group B, that is the treated group. And you have to randomize your samples. That means you have to take subjects of different sexes, ages, ethnicities, etc. Otherwise, if you end up with a lot of people over the age of 65 taking the vaccine, you won't see the real effect, right? But then in other biological research, such as bacterial extractions that I'm doing, for example, it's pretty straightforward. So if I have 3 different strains, I'll have the control and the treated strains for every single one, right? Cool. So scope of your study. You need to know, you need to go to literature and summarize any information you can have about the study, about the cells that you're going to use or organism, etc. Define the most important questions and define the missing information. So if we take as an example our data set that Daniel will provide, we aim, as he said, to compare the different instrumentation and their performances throughout a lot of labs. And they took this simulation of algal bloom. I had no idea what that was, so I did a little bit of literature research about this bacterium, Synechococcus species that we extracted. Did I say that? Okay. That causes apparently harmful algal blooms, and algal blooms are very toxic for the water, and they could kill fish and disrupt the environment. So what kind of omics strategies we're going to do? You guessed correctly, we will do untargeted metabolomics. That's why you're here. And we will use ultra high performance liquid chromatography, mass spectrometry, and of course, acquire also MS2 data, or at least the samples that we will be given already have that. And the instrument is the ThermoQ Exactive. And then define the missing information. Of course, the missing information is what is the total number of metabolites that we can detect through LCMS or through the different instrumentation anyway. And also, I guess, the different concentrations of algal bloom, what difference do they have between their metabolites? Cool, so just to be on the same page, and since I'm the first one to present, I will quickly define what is untargeted metabolomics. So it's the analysis of the whole metabolome, or as much as we can anyway, of different biological matrices. That's human, plant, bacterial, fungal, anything really. And it could be intracellular or extracellular or both, depending on your experiment and the scope of your study and your sample, of course. And talking about metabolites, we're talking about small molecules that's not that small, but they usually have a molecular weight below 1500 Daltons, with some exceptions, of course. Right, so how can we calculate the sample size before we go inside the lab and take our samples and start analyzing and sample prepping, etc.? So we can estimate the sample size through what we call power analysis. And power of our experiment is actually the probability of a true effect being statistically significant. And it depends on the effect size of our experiment. That means the relationship between the measured phenomena, the significance level, which is the risk of getting false positives, basically, or falsely identifying truly negative results as statistically significant. And then finally, the sample size. But in reality, a lot of labs actually take the size of their samples is dependent on their money, basically, and their resources, the people, the lab equipment, etc. But sometimes you can actually save money if you pre-calculate your samples. And also there's people that work with mice, and ethically you might raise a lot of questions if you start killing mice without a plan, right? Without a real justification. So, yeah, so power calculations are based on the principle of hypothesis testing, and hypothesis basically is a statement of a population parameter, right? And we have the null hypothesis, which means that there is actually no statistically significant difference between the samples, and we have the alternate hypothesis, which is the opposite. And your experiment can go 4 ways. You could accept the null hypothesis when it's true. You could accept the null hypothesis when it's actually false, where you have a false negative. Then you can reject the null hypothesis when it's true, and you can reject it when it's not true. So false positive, and I will be very clear now. So these will define errors type 1 and 2. So the risk of falsely identifying a truly negative result as statistically significant, that means a false positive, let's say, is error type 1, or we will call it A, which means that is the significance level, one of the things that our power of our experiment depends on. And then the risk of falsely rejecting truly positive results as non-significant is error type 2, or B, and the power of our experiment equals to 1 minus B. And the question of the experimental design and the power calculations especially is how to minimize both errors. Anyway, this is our goal, right? That's why we calculate our sample size beforehand. And actually, when we work with metabolomics data that are so highly dimensional, I mean, you've seen that sometimes you might have 10 samples and 10,000 features, right? This is pretty crazy. And all this high dimensionality increases the risk of getting a false positive. So it increases the risk of falsely identifying truly negative results as statistically significant. So how can we calculate our sample size? Again, we need the level of significance that's usually 5% or 0.05. That means there's a 5% risk of identifying something as identifying a false positive. Now, there's this Bonferroni correction that our statisticians suggested, Philip actually, that it corrects for this high dimensionality. So actually, if you have, let's say, 5 or 10,000 features, you can divide your p-value, which is the 0.05, by the number of features, which corrects, for this higher risk that we have in highly dimensional data of getting a false positive. Then we have the statistical power that's usually around 80%. That's pretty realistic. And then the effect size. So there you have to talk to your statistician again So if you have two groups that you want to compare, you might want to do a t-test. If you have more groups, there's different statistical tests that you can perform. But for a t-test, the effect size can be represented by the difference of means between the sample groups. And I will give you an example to clear that out. This is something between a made-up example and something that I found in a publication. So let's say our null hypothesis here is that there is absolutely no difference in the urine samples of group A, that is a group that suffers from phenylketonuria, from a group that is healthy. And phenylketonuria is an amino acid metabolism disorder. Specifically, people cannot metabolize phenylalanine. And it's pretty dangerous, actually. And actually, when you're born, you get a test on your heel immediately, like they pinch you to test it. Otherwise, you can't really breastfeed. All right, so let's say that our level of significance is, before we go there. So exactly because the tests are pretty invasive. In general, blood tests are invasive. There was a group that tried to test urine and see whether they had difference in this compound called PAG. That's a marker for phenylalanine. Okay, so we're going to compare the difference between, again, the phenylketonuria population and the health population. So let's say the significance level is 5%. The power of our experiment is at 80%. And then we need to search the literature, and especially that paper that found this biomarker called PAG. So these the authors gave out data about the concentration mean between the control group and the group that is sick. Also, they gave the standard deviation of their measurements. So we can calculate delta, which is basically the concentration mean divided by the standard deviation and the difference between the different groups, right? And this equals to 0.8. This is quite a significant difference. Actually, it's okay. And then I went into GMPS actually because I had no idea how many metabolites urine has or how many we can detect with LCMS. And I found a few groups that have deposited human urine samples with LCMS. And I figured that it's around 500, maybe 700 features per sample, somewhere there. If we have any more experienced people with urine, let me know if this is wrong. So if we use the Bonferroni correction here, we can correct this level of significance and divide the p-value by 500, which is the hypothetical number of features, average number of features that we will have in our samples, and finally get 0.001 as a risk to get false positives. So then you can use R, you can use Python, you can use your web browser. There's a lot of tools that if you give these three different numbers, it will give you the ideal number of samples that you need per group. So in this case, we have around 42. But let's say if we hire the effect, let's say the delta was 3, so the difference between those concentrations of PAG was way larger, you can see how the number is dropping significantly. So they're quite opposite. Right, so a lot of things can go wrong in your metabolomics experiments, from sample preparation, separation, detection, and analysis. And as I mentioned before, you have to use the same protocol, have reproducible results, make sure that you follow good storage conditions, good growth conditions, UV light protection. Just make sure that everything that goes on your hands anyway is not changing and is not risking the metabolites. So besides that, of course, it's very, very important to use biological replicates and technical replicates. Before preparing for this summer course, actually, I thought that using 3 replicates was fine, and that's how many I used in my experiments. But there's a lot of papers that really say that 4 minimum, you have to use 4 minimum. Anyway, so I can't redo my experiments, it's too late, but not too late for you. Yeah, again, it's very different. Biological replicates are very different from technical replicates, right? In technical replicates, you have one sample that's already grown and you just re-aliquot it in different tubes. And then in biological replicates, the starter is different. So you grow the same strain, let's say, in different vials at the same time, same conditions. Cool. Then we have process blanks, where these are samples that have no biological replicate and that will go through all the sample prep procedure. So then you can remove all the background, let's say, maybe of your media or of any solvents and that you will be using that might have carryover or yeah, some noise. Then in the part of LCMS, it's important to start always with a blank gradient. That's in my lab, for example, they just use a little vial of milky water that they use fresh milky water every now and then. And they just inject using the method that you will be using in your data later on. And then all the solvents from the mobile phase goes through and you can see whether you have any contamination, right? So then you have to maybe change the solvents, clean your columns, etc. Then they use this QC sample that you can, system suitability QC, that's what stands from SS for SS. So usually you can purchase that from a vendor and it's just a mix of standards that could actually be isotopically labeled. No, that's the internal standard, my bad. So it's a mix of standards that's basically different metabolites over a very high range and very different polarity. So you can actually test the performance of your method. Well, then now it's the internal standards. That could be isotopically labeled compounds that you can inject throughout all your you can just aliquot in every single tube before you analyze them, but they're not always applicable, especially in untargeted metabolomics. And then instead of that, we can use these pooled QC samples. Now, what they serve for, both of them, is to check for any drift, either mass discharge drift or retention time drift within your data. And a pooled QC would be basically just all your samples pooled in one vial. Then you can sub-aliquot that vial in different replicates and just inject them throughout your sequence quite a few times. So let's see an example here. I stole that slide from Martin, who was inspired by this paper. It was one of the publications that we actually suggested that you go through during your train ride or plane ride. It's a really great paper. This is Broadhurst Metabolomics 2018. That's the latest one. All right, so here they have a pooled QC protocol for biofluids. That's the ideal protocol, where a biofluid, by biofluids we mean blood, urine, plasma, anything that's liquid. So here they have two different groups, 5 replicates per group, biological replicates, and they take 10 microliters from each plasma sample and they pool it in one tube. Then they go through all the sample preparation procedure, extraction procedure, and then they re-aliquot the pooled sample in 3 different vials. That's just to have technical replicates. And then you can inject the pooled QCs from 1 up to 4 times per vial throughout your sequence. Then you can monitor for any mass discharge or retention time drift. And here, this pooled QC can actually reflect the variance you have through your sample preparation technique. But if you have cellular or tissue samples, you can't pool them before the sample preparation technique because the samples are not homogenized. So you have to homogenize, do all the sample prep, and then pool them. So that will not reflect for the variance you have in the sample preparation technique, right? It will only reflect the variance you have in the acquisition part later on. All right, so choosing a sample preparation protocol, or the perfect sample preparation protocol. If you are doing targeted metabolomics, you might have a perfect preparation protocol. If you know your metabolite, if it's polar, non-polar, has a lot of carbonyl groups or amines or amino acids, anything, then you can more or less higher the yield of extraction and then optimize your sample preparation. And you can see here, this is a really nice figure from Eva, a PhD student of Martin, where with increased polarity, you just use different techniques like normal phase, reversed phase, ion chromatography, etc. But in non-targeted metabolomics, of course, there's no perfect protocol. There's always sacrifice every step of the way. You might lose some metabolites. If you choose LC, of course, you might lose your volatile metabolites. Yeah, so keep that in mind. Of course, in your wish list, you should remember to quench your sample when you begin, right before the extraction. You should quench. That means you should stop for any enzymatic activity that's happening in your vials, in your samples. You should be careful to have the simplest protocol as possible in the fewer steps as possible, and then you can avoid contamination. And of course, you know that every step of the way you might lose some metabolites. So let's say we dry and we dissolve quite a few times in different solvents, then you just leave behind more and more and more things. Cool. Then the third part is having reproducible data. Make sure that if you do the same experiment a week later, you get the same data. That's very, very important. And of course, keep the protocol saved somewhere, every single step of the way, very, very detailed. And then finally, of course, the holy grail, get as many metabolites as possible. So in one example of metabolite extraction of biofluids is here. Again, I stole that from Martin. So here is a sample of blood that if you centrifuge, you end up having plasma, white blood cells and red blood cells. And then here they extract the biofluid with 3 parts of methanol and 1 part of sample. Now you see that there's no different step really between quenching and extracting here, because solvents are used for both and methanol can be used for both. So then of course they shake the sample, they centrifuge it, and they dry it using gentle nitrogen stream, which I highly recommend if you're drying. This is, well, small samples. If you dry like liters, then of course use a rotary evaporator. And then redissolving and moving everything in an HPLC MS vial. Now, an optional step that actually I'm personally using with my data, because I'm working with 24 and 96 well plates, so a lot of samples, like thousands, is a solid phase extraction. Especially here in the figure, we're looking at a 96 well plates that has in every single well a sorbent that basically is the same thing as you can find in an HPLC column. It could be any chemistry in there, carbon 18, carbon 8, just silica, anything depending on the chemistry. So what you do is that you condition the columns, you load your samples, you wash with water, and you elute with methanol. Usually that's the standard protocol, and you take out this eluent anyway. Now for my data, and I think if you test that, you will be also positively surprised that it helps a lot with removing ion suppression from your MS data. But you will talk about it a little bit later. Some of you probably know what I'm talking about. So usually I am able to remove salts and some sugars from my media that's pretty nasty, and then really my data looks so much better in the end. But of course, solid phase extraction could be also for single data. They also sell just single columns that you can use, just way less data in general. Cool. Then this is a really nice paper by and figure, especially by LC, that shows the whole metabolomics workflow, just from very diverse samples to quenching, extracting, separating, analyzing, and then just acquiring all the data. And really, they summarize here everything that we said today and even more that Martin and Daniel will talk about. So what to be careful in the part of chromatography and mass spectrometry, and what we covered today, what to be careful for sample preparation, randomization, extraction, etc. And you see that he says here that you should at least have 4 replicates. So technical and analytical, he says, are worth of consideration. Yeah, I highly recommend to use technical replicates. I will not stay here because half of it is just irrelevant to the presentation, but it's just a really nice figure of the whole metabolomics workflow that most of us are doing in the lab. So in summary, experimental design can save you time and money and save you from raising ethical questions in some cases. Of course, there's no perfect sample preparation protocol for untargeted metabolomics, but try your best, and there's always literature review, there's always method development that you are going to have to go through in the lab. Cool. Any questions? Yeah.
You go first. My problem is always like when I grow bacteria, that they're very dynamic and sometimes very hard to get the same results twice.
Yeah.
So, and especially if you have pigmented bacteria, they drive me nuts. So if I want to do like an untargeted approach, it could be just the condition they're growing and changing so much that I don't get a reliable result at the end. So is it not possible to do something like this, or what would be your solution? Or is there no solution, or is it not possible?
What bacteria are you working with?
It's a gram.
So I think, no, there's not much you can do, I guess, but maybe using a lot of biological replicates would be the way to go here, as many as possible. Yeah, and of course, you're extracting at the same time point and everything is just...
Exactly, but there's no like very small variation in like humidity in the labs or something already trigger them differently, and then they have different.
So that's another thing. Yeah, we also had the same problem in the lab. Sometimes the incubators were just not behaving. Some person would come and like change the RPMs in the middle of my yeah. You just have to be a bit careful with the, maybe use the same incubator all the time if that's possible. Really, that's, yeah. Is that no? I can see
we try like, but that's something I would like to try as well with those samples. See, okay, the changes all the time, but like what stays the same.
Yeah.
There's just a few molecules which behave.
Yeah, there could be just the same pathways that you see, but in different time points, right? Just shunt products or some products before the end pathway. Yeah. I maybe somewhere else can suggest something. I feel you, but
yeah, I think it's a very important point to bring up. I would just reiterate. So I think what you said is totally on point. I would increase the sample number, right? And then, I mean, if you have like huge variance and you go back to the power calculation, if you show, then that would just mean that you need more replicas.
True. Yeah.
And then the other thing that might be a very straightforward practical solution is get the replicates from like the same moments, right? So like you eliminate all the environmental factors like humidity and temperature if you grow them in parallel. And then I would expect hopefully less variance. And if you have like, I don't know, 20 petri dishes from the same incubator at the same time, then maybe that's a better design.
We can do the same thing on like we have different plates and we take samples on like different plates, they look completely different.
Okay, wow.
They grow like, but this is like this two bacteria I know with this. We want to understand why and like why they are like so variant. What's the reason behind it? So this is what I would like to try.
So that's on agar.
That's agar and marine agar.
They can be highly mutated on the plates also. If you see phenotypically, go for the ones that look identical. Yeah. Sometimes we have that with actinomycetes, they just mutate on the plate and some colonies look slightly different. So you want to go with the ones that are really phenotypically identical. Yeah. In that case, same frozen stock also very important.
Yeah. Okay. Now we are measuring per milligram how much agar we put on the plate so that they're different. So it's a nightmare, but very interesting.
Have you tried liquid or?
Yeah, we grow them, but they don't produce this pigment when we grow it on liquid.
Okay, yeah.
And so they are
So you need to suffer.
tricky. Thanks.
Good luck.
I had a more general question about the replicates.
Mhm.
You apparently struggled with this yourself as well.
The 3 replicates?
Well, what's a biological replicate? What's a technical replicate? And how many should you include? Is there maybe someone in the room knows, or maybe you know?
So what is the
What
difference
is-
between the biological and the technical replicate?
Well, yeah, when because you said, for instance, use the same strain growing 3 times.
Yeah, because I guess during growth you have some variation, right? I personally, if I grow actinomycetes, I'll have just different really colonies and different cells. So you want to grow them in just different flasks and monitor their growth, and then technical replicates would be after quenching. So if you have one vial, one flask, and you quench it, or do the whole sample prep, whatever you want to monitor. But the technical replicate would be if you stopped all the enzymatic activity and then re-aliquot. Maybe some, yeah.
On the proton side, the biological replicate is a variation that is generated through the biology, through the sampling. The technical replicate is a variation that is generated by the limitation of your instrumentation. Because of the way you acquire data, especially
Yeah.
part of the
Exactly, or the sample prep. If
No,
you-
I get that. My question was not very clear. Where do you draw the line between where do you have to start from technical, for instance?
You
Because
always have to.
the technicality starts, you say with quenching, but it can already start with basically sampling just before the quenching. And the same is true for biology. You said same strain.
Yeah, you can't split the cell.
In my PhDs, we were always adamant you had to generate at least 3 independent mutants, and those would be the biological. So same mutants, same, but then those would be the biological mutants. So
Yeah.
there's quite a big difference between what people mean with biological and
Oh, for sure.
And of course,
Yeah,
because
maybe.
we don't work on the same stuff. But I'm wondering, is there some rule for it?
True, I would say, yeah, I can completely see your point from just taking cells from the frozen stock, I guess. You could start from there. Maybe that would be the right way to go. So just grow those frozen stock cells in 3 different plates or 3 different vials. Yeah, probably.
So it's a really interesting question.
Yeah, it's really
I
interesting.
would be curious what people in the audience do. So like for me, and that's totally subjective, right? But for me, technical replicate means actually replicated injection to the mass spec, which is like very low and very conservative. Unless you do things like DDA, MS of very complex samples, right? Then actually you have a huge variance on MS2 level, not so much MS1. So what you just said, like we would, just like process like all biological replicates like independently, but not do like technical replicates there. But that's just one way of doing it. So I don't know, like who in the lab, who in the audience considers a technical replicate to be like a replicate? Can you raise your hand? Okay, so that's what most
Most of the people.
Okay.
I mean, then you only capture the technical measurement error.
Yeah.
But you also have a technical extraction error.
Exactly.
So I think you have to split it up. The technical replicate can be different things. It can be the technical measurement error, which is what you're seeing.
Yeah.
Repeated injection of the same thing. It can be technical extraction plus injection, because you always get this as well. If you have exactly the same material and do the extraction and then inject it again. So I think you have to split this up.
Then your samples go up exponentially.
Yeah.
So that's exactly what my question was about. Where do you decide what you decide is a biological and technical replicate? But there is no.
Well, I think the good thing is if you know your system, at some point you can say that your extraction error is much larger than your really technical measurement error. So you can pull
What?
and you can say, okay, I'm going to work with
This one?
So,
The
so you do both then?
Yeah.
Replicas?
Well, no, we've done the real measurement replication often enough that we know it's much smaller than our technical one, and that's why the extraction one, and that's why we only use the replication from the full process.
That's interesting. Maybe one additional, can I just add one thing? Maybe an additional very important aspect there is like practicality. Right?
Yeah.
So depending on what scale you work, you may do not want to have multiple technical replicas, for example.
true. That
So
would increase insanely then.
Yeah, so the technical measurements, that's the repeated measurements on the platform, whatever platform it is. And if you want to have the sample prep included in that, then go with this approach. And maybe that was the other approach with tissue. Maybe sometimes it's, as Evie said, it's not possible always to have the pool sample before sample prep. Then you will go with this approach. So, and we will look deeper into that in the next session after the break. So maybe, but please, questions is good. Just also ask, I would recommend to ask questions during the talks. You're very welcome to do that.
Any other questions, or should we have some coffee? It's a trick question. Oh.
now it's